Friends, I am Dr. K.D. Mishra and today we will discuss blood groups and transfusion of blood. As you know, in different conditions, especially in case of accident, in case of anemia, and in case of certain other medical conditions, a person has to be given blood. We call it blood transfusion. And for that, it is very important that one must have the complete knowledge of blood group typing and compatibility of blood for the patient. Now, first of all, let us see some of the terms which are used in the blood group testing. That is, one is agglutinogen. Agglutinogen, you always remember the word gen. That is for antigen, a specific kind of protein which is present on the surface of RBC. The second one is the agglutinin. This one. Agglutinin is the antibody which is present in the blood plasma. Now, when a particular antigen is present on the RBC and a particular antibody for it is present in the blood plasma, then a reaction occurs between these two, thereby hemolyzing the RBCs, and this is called as agglutination. So, a specific reaction it is in which a particular antigen clumps in the presence of its specific antibody. That means if the antigen A is present on the RBC and antibody A is present in the plasma, then agglutination will occur. So, these two cannot be found together on surface of RBC and in the plasma. Now, there are hundreds of antigens found on the surface of RBCs. You know, the proteins are the basic component of antigen. So, since the surface of blood cell is made up of protein, one of the component is protein. So, those antigens are present on the RBC, but not all of these are important from the point of view of blood transfusion. Because most of these are weak and usually they are used only for the purpose of inheritance. That means in certain cases, when you want to see the inheritance of a particular gene from parents to the child or in a particular community, it can be used for the sake of study of evolution of human race or that. In those cases, these antigens are studied. But two types of antigens are important for the purpose of blood transfusion. These are ABO system and RH system. That is, antigens and antibodies of ABO system and antigens and antibodies of RH system are important from the point of view of blood groups. Now, the blood group of an individual is determined on the basis of the antigen which is present on the surface of RBC. When a particular antigen is present on the RBC, corresponding antibody should be absent in the plasma of that individual. That means if a person is having antigen A on its RBC, antibody A or alpha cannot be there in its plasma, because if it is there, it will cause agglutination. And if the antigen B is present on the RBC of a person, antibody B cannot be there in the plasma because it will cause agglutination. So, this forms the basis of blood group identification and blood group transfusion. Now, there are about 25 blood groups which are due to about 400 antigens. Now, apart from ABO and RH system, the other system which are used for medical purpose and medico-legal purpose, these are MNS, Levy's, Duffy, Kell, P and Lutheran system. Now, what is this medico-legal case? You know, in case of any murder, or in case of rape, or in case of paternity test, or in case of maternity test, there are certain legal issues. For settlement of those legal issues, these antigens of minor importance from the point of view of blood transfusion are being tested, and they are legally accepted in the court of law. Now, in ABO system, first of all we will deal this. In ABO system, there are two antigens present on the surface of RBCs, antigen A here and antigen B. In antigen A can be of two subtypes. It is antigen A and antigen A1. The genes of ABO antigens are located on chromosome number 9. These antigens are chemically oligosaccharides. A antigen has a terminal sugar, that is N-acetyl galactosamine, whereas the B antigen has galactose. Now, agglutinins or antibodies which are found in the plasma are antibody A or alpha and antibody B or beta. Similar to antigens, the antibody A are also two types. One is A proper and another is A1 or alpha proper and alpha 1. Now, just to avoid confusion in these alphabets, for antibody alpha and beta is used, and for antigens capital A and capital B are used. Antibodies are always found in the plasma. ABO antibodies are naturally occurring. This is the important character which is important in blood transfusion. When we say naturally occurring antibodies, that means they are present since birth. If a person has antigen A, he cannot have antibody A, but he has antibody B, which is naturally found in his blood. Now, Antibody A or alpha and antibody B or beta, these are agglutinins which are chemically globulin fraction of plasma proteins. This is a IgM type of antibody. Karl Landsteiner was the first to identify or discover the blood groups A, B and O, not AB. So, he was the first to discover these three blood groups A, B and O. Therefore, he is honored as father of blood groups. Later on, De Castello and Sterle in 1920 discovered another blood group of ABO series, that is blood group AB. Now, the definition of blood groups and their identification is governed by a law called Landsteiner law, which states that a particular antigen cannot occur with its corresponding antibody, or if a particular antibody is present in the plasma, the corresponding antigen cannot be there on the surface of RBC. Let us see, the law states that agglutinogen, that is antigen, and corresponding agglutinin, that is antibody, are never found together. In other words, if a particular agglutinogen is present on the surface of RBC, the corresponding agglutinin or antibody must be absent in the plasma. Conversely, if a particular agglutinogen is absent on the red blood cell membrane, the corresponding agglutinin must be present in the plasma. The gist is that since a particular antigen, when it reacts with its corresponding antibody, agglutination occurs. So, these two cannot be found together. But in case of ABO system, the opposite antibody, that means in case of antigen A on RBC, so antibody B is present in the plasma of that person. So, opposite antibody is found in the plasma. This law or this formula does not apply to the RH system, because if RH antibody is present on the RBC, RH antigen is present on the RBC, antibody for RH is not present in the plasma. But if antigen for RH is not present on the RBC, as per ABO system, there should be a antibody for RH, but it is not present naturally. However, if a RH negative person is given RH positive blood, then what will happen? Since it is a new protein for that person, antibodies will be formed for the RH antigen. So, antibodies will be developed and the person will learn how to develop such antibodies for future, but they are not found since birth. Now, here in this table, you can see different blood groups, then presence of antigens on the RBC and presence of antibody in the plasma. In case of blood group A, RBC has antigen A and the plasma contains anti-B antibody, or we can call it beta antibody. In case of blood group B, RBC have antigen B and the plasma contains anti-A antibody, that is alpha antibody. In case of AB, this is a character which is governed with a law of inheritance called codominance. Both these genes of antigen A and antigen B are found together, and both these antigens are found on the surface of RBC. So, these two antibodies cannot be present in the plasma, because if any one of these is found, the agglutination will take place. Now, in case of blood group O, I always say blood group O means 0. That means 0 antigen for this ABO system. Such a person do not have any antigen A or B on the surface of its RBC. So, the opposite antibodies must be present in the plasma. Therefore, in the plasma, both antibody A and antibody B, that is antibody alpha and antibody beta, are found in the plasma. Now, blood group typing or blood group testing. For blood group testing, we use antisera, or singularly called antiserum. A small amount of blood is taken and it is mixed with antiserum A and antiserum B for identifying whether the blood group is A or B or AB or O. Now, testing a blood group of ABO system is very important for matching of blood sample of the donor and the patient. If it is mismatched, then severely it will affect, coagulation will occur in the circulatory system, and there could be a threat for the life of the patient. So, what we do is we take antisera singularly called antiserum A and antiserum B. Okay. What we are doing is that we mix a little amount of sample into this antisera A and antisera B and see, observe the agglutination under the microscope. See, this is a slide here and this is another slide here. This is another slide here. Now, we have got two antisera. One is of blue color like this, and another is yellow colored Now this is B and this is A. Now we will prick the finger of the donor or patient, and we take out drop of blood, and then those drops of blood are mixed separately into antiserum A and antiserum B in slide. Now what will happen? See, there can be two possibilities. Either in the drop the blood cells will look like this. They look without any clumping, without any agglutination like this, or there can be agglutination and this blood group forms a mesh like this. This shows a reaction between antigen and antibody. If same antigen and antibody are mixed, such type of reaction is seen. Now we see different conditions. If agglutination is seen here, that means this is blood group A. Okay. If agglutination occurs here in antiserum B, that means blood group B. If agglutination occurs in both, this is blood group AB. And if agglutination does not occur in either A or B, it is blood group O. So that is how the blood group of ABO system is tested. Now the same experiment which I just explained is shown in this table. A person with blood group A, when it is tested, it gives a positive reaction in antiserum A and negative reaction in antiserum B. In B blood group, there is a negative reaction in antiserum A and positive reaction in antiserum B. In case of AB, a positive reaction in both antiserum A and antiserum B. Such peoples are called universal recipient because they do not have antibody for either A or B. In case of O, since it do not have antigen for A and B, the reaction is negative in antisera A and antisera B both. These are called popularly as universal donors because they do not have any antigen either A or B. Now the second system, which is very important for blood transfusion and genetic purpose also, that is the RH system. This is due to a factor which is found on the RBCs popularly called as antigen D. It was first described in the rhesus monkey by Landsteiner and Biener in 1940. There are 6 antigens for the RH group, that is denoted in the capital letters like C, D, E and small letters C, D, E. Out of these, the most effective antigen is called antigen D. Therefore, for the sake of studies, it is always denoted as RH positive antigen or antigen D. Now, how it is different from the ABO system? We have seen in ABO system that there are naturally occurring antibodies present in the plasma. But in case of RH, no naturally occurring antibodies are found in the plasma. However, they can develop when RH antigen enters into the blood of a person with RH negative blood group. So, it is called anti-D antibody. It is IgG type of antibody and it is most reactive at the body temperature. As you know, our body temperature is constant. We are warm-blooded animals. So, this is called as warm antibody. So, once it is formed, it is more reactive at our body temperature. Second thing which is important is that this is antibody which can pass the placental barrier and hence it can pass from the circulation of mother into the circulation of fetus. There are two groups in RH system. If antigen D is present, it is RH positive. If RH antigen is absent, it is called as RH negative. Now, the occurrence of RH system in various populations, human populations in different parts of the world, they are among Indians 95 to 98% peoples are RH positive, whereas only 2 to 5% peoples are RH negative. In case of whites, 85% are RH positive and 15% are RH negative. In case of American blacks, it is similar to Indians. About 95% people are RH positive and 5% are RH negative. But in the African blacks, this gene of RH positive antigen is very common in the population and therefore, among them nearly 100% peoples are RH positive. Now, if we talk about the inheritance of RH antigen, you know, the RH antigen is a comes from a dominant gene. When we talk about dominant gene, we always represent it by a capital letter. Now, this gene is located on chromosome number 9. We have seen that the genes of ABO system are located on chromosome number 9, but the genes of RH system are located on chromosome number 1. It is a dominant gene. So, RH positive people can be homozygous or heterozygous. They can be either or they can be heterozygous. Both of them will be phenotypically RH positive. So, when a RH positive person marries to a RH negative lady, so there are chances of having a RH positive child at least 50%. If it is dominant, then 100% childs will be, chances of RH positive will be 100%. If it is heterozygous, then there will be a possibility that the fetus will be Rh positive by 50%. Now, nearly 40% population of Rh positive people is homozygous, whereas a more share of people are heterozygous. So, there are always chances of a Rh negative female may conceive a Rh positive child. Now, same thing is given here. If one parent is homozygous Rh positive, there all the children will be Rh positive. That means if the male is Rh positive and female is Rh negative, there all the children will be Rh positive. If both parents are heterozygous for Rh, the chances of their children having Rh positive will be 75%. If both are Rh positive and heterozygous, so the chances of being Rh positive will be as much as 75%, and only 25% chances are there of child being Rh negative. But if 1% is heterozygous Rh positive and other is heterozygous negative, then the chances are 50% being Rh positive, as I just mentioned. So, if the female is Rh negative and male is Rh positive, at least 50% chances are there of their children being Rh positive. If the both parents are homozygous for Rh negative, then obviously their child will be Rh negative. Now, Rh incompatibility. You know, when there is a mismatch of Rh group, if a person of Rh negative blood group receives a Rh positive blood, then it is called as incompatibility. If it is given first time, if a Rh negative person receives Rh positive blood first time, then there will be no serious consequence to that, as the formation of Rh antibodies takes a long time. They are formed very slowly and it takes about 2 to 4 months to develop a significant titer of antibodies that can cause harm to the recipient. Since the donor's RBC, now you know, RBC comes from donor also. They are having Rh antigen. So, if the recipient develops antibody, there can be a reaction. But since antibody formation takes about 2 to 4 months and the lifespan of donor's RBC is about 120 days. So, by the time the sufficient amount of antibodies are formed in the recipient, by that time the RBC of donor in the recipient, they are already hemolyzed. Now, when a Rh negative person receives a Rh positive blood and antibodies are formed in response to antigen D, such a person is called sensitized. If subsequent transfusion is given of Rh positive blood, then severe transfusion reactions may occur, leading to the death of the recipient, death of the patient. This is similar to mismatched transfusion of ABO blood group system. Similarly in ABO blood group system also, if a person having antibody B, that means if a person of blood group A receives blood group B, then the RBCs of donor will be agglutinated in the recipient's blood, causing severe consequences. Now, another important condition in which there is a incompatibility of RH between mother and her child present in the uterus, so intrauterine life. It is called as erythroblastosis fetalis. That means the damage of blood cells, RBCs, in the fetus. It is also called as hemolytic disease of newborn. That is, hemolysis occur in the newborn. When RH incompatibility in newborn, it is called as erythroblastosis fetalis. It is seen when husband is RH positive and wife is RH negative. So, as I just mentioned, in that case, there is every possibility, at least 50% possibility is there, that she may have conceived a RH positive embryo. Now, what happens? As mentioned earlier, these RH antibodies can pass the placental barrier. So, if these antibodies passes from the placenta to the fetal circulation, hemolysis of fetal RBCs takes place. That is caused as hemolysis, hemolytic disease of newborn. Now, generally, first conception, if such mismatch happens, that means if for the first time a lady conceives a RH positive fetus, since that lady naturally do not have RH antibody, so there will be no harm to the first child. But what happens? During parturition or childbirth, there is lot of bleeding from the uterus, and some of the blood cells of fetus passes into the maternal circulation, and those cells generates a sensitivity by that RH antibodies are produced in the lady, and now it is called sensitized. Now, if she conceives a second child who is also RH positive, then in that case, from the fetal circulation, the RH antibodies passes from the maternal circulation, the RH antibodies passes into fetal circulation, causing hemolysis of fetal RBCs. So, in the successive pregnancies, it will be harmful. Or even if that lady is given RH positive transfusion earlier to first pregnancy, and if she is sensitized, then her first child will be affected by erythroblastosis fetalis. Just I have explained the same thing, that when the body crosses the placental barrier, then there is a rapid production of RBCs in the spleen of the liver and spleen of the fetus. As we have seen in the embryonic condition, in the mesoblastic stage, the blood cells are primarily formed in the spleen and liver. So, many of the erythroblasts are produced there. In this process, many immature nucleated blastic cells passes into the circulation. Therefore, it is called as erythroblastosis fetalis. In severe cases of erythroblastosis fetalis, usually death of the fetus occurs, but in some cases, if death does not occur, then the child suffers from severe anemia. Now, what happens due to this impairment and damage of RBC? A lot of bilirubin is produced, and that is circulated to different areas of the brain, mainly motor area of the brain, where it precipitates, and this condition is called kernicterus, where the bilirubin is accumulated in the brain. Now, treatment of erythroblastosis fetalis. Now, there can be, if it is diagnosed that the mother is having Rh positive fetus, then there can be a blood transfusion to avoid the lysis of blood cells. There can be exchange transfusion in newborn. You see, the blood of neonate is replaced by Rh negative blood. As just mentioned, in erythroblastosis fetalis, the embryo has Rh positive blood. So, for treatment of that, Rh negative blood is regularly transfused into the newborn at the rate of 400 ml after every 1.5 hours, and it is repeated several times as long as 6 weeks. By this time, the antibodies which have passed from mother to the fetus and are already there in the circulation of child, they are destroyed, and then the blood is replaced by the Rh positive cells. Now, for treatment, this is the most common treatment of the childs which are suffering from kernicterus and this destruction of RBCs due to loss of blood and paleness in the blood, jaundice-like symptoms. So, they are given a treatment of phototherapy. As when the patient is exposed to the light, the bilirubin undergoes photooxidation and polymerization, making it less toxic. Now, for prevention of erythroblastosis fetalis, the mother is prevented from being sensitized for Rh antigens. For that, immunoglobulin, that is anti-D antibody, is administered to the expected mother 28 to 30 weeks of gestation. That means if the mother is having anti-D antibody, then when the Rh positive cells of the fetus will enter into the circulation of mother, they will be immediately destroyed, and mother will not learn how to produce anti-D antibodies. Then again, anti-D antibody is also administered to a Rh negative woman who delivers Rh positive child to prevent sensitization. As I just mentioned, during the parturition, some blood cells of the fetus enters into the maternal circulation, thereby making the mother sensitized for antigen D. So, for that, antibody D are given to the mother so that the cells, Rh positive cells which enter into the maternal circulation are immediately destroyed, and it prevents the mother from being sensitized for Rh. Now coming on to the second part, that is blood transfusion. In medical practice, transfusion of whole blood or a component of blood like RBC concentrate or plasma is very common. Commonly, transfusion of whole blood is done, but in severe anemic conditions and in cases of severe hemorrhage, even RBC concentrates are transfused. It is very important for a physician to know the indications, the methods and hazards of blood transfusion before transfusing blood to a patient. Now, certain indications. In case of surgeries, when there is a possibility of lot of blood loss, then if there is a possibility of about 500 ml blood loss, then a transfusion is planned. Or in cases of when the patient have the low hemoglobin value, then also transfusion is planned prior to surgery. In case of acute blood losses, like in case of accident or any other region, transfusion of whole blood is preferred because all the components of blood are lost. So, it is preferred to transfuse the whole blood. In case of chronic anemia, where the anemic conditions are prolonged for a very long period, then it is preferred to transfuse packed red cells. Now, in case of burns, where lot of fluid is lost from the body, but the number of RBC is not that much affected, because in the burn, lot of fluid portion is lost from the body, and to compensate that, the amount of water in plasma is reduced, and for that, plasma and colloids are given to maintain the blood volume. Now, in case of purpura, purpura is a hemorrhagic condition in which leakage of blood occurs from the blood vessels and it is accumulated under the skin. It is primarily because of the insufficient number of platelets, and therefore transfusion of platelets is done. Now, in case of hemoglobinopathies, that means the defective synthesis of hemoglobin, and because of that the amount of hemoglobin is less. In those cases, repeated multiple blood transfusions are required to maintain the concentration of hemoglobin and to inhibit erythropoiesis of defective sickle cell. Like this is one example, sickle cell. Sickle cells are produced due to defect in the beta chain of hemoglobin. It is a point mutation. So, such cells which are not circular, they are sickle shaped and with defective hemoglobin, which is denoted as sickle cell hemoglobin, is produced. So, Neither the cells are competent to withstand the osmotic pressure, they are destroyed, nor they have a normal hemoglobin. So, there is a deficiency, we call it sickle cell anemia. So, repeated transfusion of blood in case of thalassemia and in case of sickle cell anemia is given so that the oxygen level in the blood is maintained and the production of RBCs is not stimulated in the bone marrow. Now, procedure of transfusion. A procedure is called autologous donation. This means the blood of the patient is collected and it is reinfused in the same patient. This is called autologous donation. Now, it has got certain advantages and disadvantages also. If there is a planned surgery, suppose the surgery is not urgent and a person is to be can be operated after a month or so, his blood is withdrawn and it is kept in a blood bank, and then when during surgical procedure blood is needed, the blood of the same person is reinfused into it. It avoids any of the risk of the any of the risk of the donor's blood having any kind of diseases like AIDS or any other communicable or any other transferable diseases through the blood. So, blood-borne infections can be avoided in that. Then allergic or hemolytic transfusion reactions can be avoided. It is safer for the people who have got rare type of blood groups like A negative, B negative, B positive, AB negative, AB positive and O, so it is safer. You just plan a surgery after some time, after 1 month or 2 months, collect the blood from the same patient, keep it in a blood bank, and then if needed during the surgery, the same blood can be reinfused in the person. This is called autologous donation. Now, it has certain disadvantages also. Although they are very few, there is a risk of anemia. If you draw the blood from the same patient, he may suffer from anemia. Then there is a risk of bacterial infection because during the storage of blood, some bacteria may develop in the blood sample. Then unused blood cannot be donated to any other person. This is one of the disadvantages of autologous donation. Now, in the transfusion procedure, there are 3 things which are to be observed and important. One is the selection of donor. A donor should be a healthy person, must have the correct composition of blood. He should not have any type of blood-borne diseases and all that. Then a blood group testing should be done for ABO system and for RH system. Even after doing this, before transfusion of blood, there should be a cross matching of the blood sample of donor and the blood of recipient, that is patient. The blood of both is taken, it is mixed on a slide, then it is observed under the microscope. If there is agglutination of any degree, such a blood is rejected. Here you can see the grounds on which donor is selected. He should be a healthy person. Blood sample be screened for transfusion transmissible diseases. That is, one is HIV, another is hepatitis, like STD, syphilis, malaria, filaria, etc. There should be no disease of this type. Age of the donor is again important, because some of the blood cells, when become old, they do not work properly, they become senile and they are destroyed very easily in the recipient blood. So, the age should be between 18 to 60 years. Again, very important criteria to be considered is the percentage of hemoglobin. It should be at least 12% in case of female donors and at least 13% in case of male donors. Again, same thing, ABO incompatibility should be checked. The ABO system, the ABO group of patient and ABO group of the donor should be matched, should be tested for RH incompatibility also. The ABH antigens reacts very quickly. So, the incompatibility reaction starts immediately. It is somewhat delayed in case of RH, but then it is also very severe. Then again, just I mentioned that before transfusion of blood, cross matching of blood sample should be done. So, as to eliminate any type of incompatibility of the antigen antibody reaction, whether for the known antigens or for the unknown antigens, there should be no agglutination in the samples of donor and the patient. As mentioned earlier, there is a common practice of using these words like universal donor and universal recipient. A universal donor is that which do not have any antigen. So, blood group O minus, that is ORH negative. ORH negative is called universal donor because the RBC do not have any antigen on them. So, it will not react with the antibodies of ABO system when such a blood is transfused into a patient. Similarly, a universal recipient is that which do not contain any antibody in its blood either for ABO system or for RH system. Therefore, the AB positive has both the antigens but do not have any antibody. So, therefore, it is called universal recipient. It can receive blood from any of the blood group A, B, AB and O. Either it is RH positive or RH negative. Therefore, it is called universal recipient. Now, second part is collection of blood. About 350 ml of blood is collected from a donor at a time, not more than that. Number two, it is collected from the antecubital brain. Number 3, it is collected in the plastic bags which contain anticoagulants. These anticoagulants include citrate, phosphate and dextrose, that is, it is called CPD. The sodium citrate binds with calcium and prevents clotting. You know, calcium is very important for blood clotting. So, for preventing the blood from clotting, the calcium is necessary. Then phosphate buffers the pH, dextrose provide energy to the RBCs, and adenine provide substrate for ATP synthesis. So, all these are present in that plastic bag in which blood is collected. Now, effect of storage. The blood sample is stored at 4 degrees centigrade and it can be used within 2 weeks. Preferably, it should be used within 2 weeks. Now, what happens during the storage time? The RBCs, they tend to lose their shape. They becomes spherical and loses membrane flexibility. As you know, we have already seen in the metabolism of RBC, they need energy for maintenance of their shape, flexibility and all that. So, due to that, they becomes spherical, the flexibility is lost and the stability of membrane is lost. But then, when such a blood is transfused in a person, then RBC regains all these characteristics. Now, here you can see the leukocytes and platelets are destroyed after 24 to 48 hours of storage. There is a depletion occurs in ATP, as I just mentioned, and the adenine content. After transfusion, just I mentioned that the RBCs resume their shape, their fragility within 24 to 28 hours after transfusion. Then nearly 80% of RBC survives transfusion if given within 14 days of storage. Therefore, it is preferred to do the transfusion within 2 weeks of sample collection. Now, transfusion hazards. If there is a mismatched transfusion due to ABO incompatibility, then an acute hemolytic transfusion reaction starts. That means the blood cells of donor are hemolyzed in the recipient. It starts immediately because of the hemolysis of donor. Then complications, shivering occurs, fever comes, then hemoglobinemia and hemoglobinuria. That means when the blood cells are broken, lot of hemoglobin is released in the blood. That is a condition called as hemoglobinemia. That means hemoglobin in blood. And when more amount of hemoglobin outside RBC is available in the blood, it is filtered in the kidney and passes from the urine. That is called as hemoglobinuria. And due to scarcity of blood, due to anemic condition, the skin, the nails, then conjunctiva of eye becomes yellow. These are the symptoms of jaundice. Now, in case of if the transfusion is not proper, then there can be acute renal failure. This is due to destruction of RBCs, release of hemoglobin. That hemoglobin blocks the renal tubules, thereby causing dysfunction in the kidney and failure of kidney. So, this is due to hemoglobin. Then hemolyzed RBC causes vasoconstriction, and sometimes it also affects blood pressure. Now, among other hazards of blood transfusion, one is the immune reaction. You see, the hemolyzed RBCs causes circulatory shock. The second one is these conditions together causes renal shutdown. The kidney stops working. We call it renal shutdown. Then sometimes massive blood transfusion is to be given, and that becomes fatal for the patient. When total blood volume is replaced or more than 10 units are given within 24 hours, it causes a load on the heart. We call it circulatory overload. Sometimes cardiac arrhythmias, that is irregular heartbeat, and cardiac arrest may occur due to high potassium content which is found in the stored blood. Some other hazards include the febrile reaction. Sometimes patients feel cold due to rise in body temperature. This is due to pyrogens which are present in transfusion apparatus. You know, when the blood is given, apparatus is used for that. It is called transfusion apparatus. Then allergic reactions. Some allergic reactions are seen in some cases like nausea, vomiting, itching. Sometimes severe cases are seen which may lead to anaphylactic reactions. That is all for today. Thank you very much.